All right, take things away, Olga and Tony.
Okay, thanks. So bear with me while I try to share my screen. Okay. And then am I in presenter mode to everyone?
Just gotta flip it.
Okay, let me flip. Swap. How about now?
That's
Perfect.
good now.
Great. Okay, great. So thanks again to Dan for organizing this entire group. Really appreciate all the work that goes into that. And um, like we mentioned, Olga and myself will be discussing a bit about the work we've been doing incorporating in silico tools to predict developability and immunogenicity during early stage discovery. So I'm just gonna a brief overview of what we'll be talking about today. So I'll just share a bit about the department, and then Olga will be walking us through Immunoscreen, our in silico immunogenicity assessment tool, and then I'll bring it back into how Immunoscreen fits into our broader platform inside where we use to manage our developability risk assessment. And then Olga will finish off with a bit more earlier stage work about modeling clinical ADA. Um, so let me just kick things off again. A bit about the department. So Olga and myself are part of biologics engineering. We're spread off across a couple of different sites, supporting both the oncology and biopharma pipeline. And actually, really excitingly, Waltham will soon become Cambridge. As soon as we are able to move into our new site in Kendall Square, once that's renovations are completed, and then very excited to also kind of build out that team at that new site to continue supporting the growing pipeline for those of you in the Boston area. And then kind of an overview of all the functional teams within the department. So while I'm in the protein sciences and analytics group, all the work I'll be talking about today is really collaboration of all the different teams here, particularly the folks in the augmented biologics discovery and design group, as well as our high throughput automation team, really meaning the challenges of bringing more advanced and so-called tools and machine learning technology into the department, freeing up resources so we can expand what we deliver on, particularly as the supporting of novel modalities of ADCs and cell therapies, which do require a lot more support as we kind of work out the pros and cons of these different modalities. So to provide some context about kind of where all these in silico tools we'll be talking about really are being deployed, my group operates mainly in the early discovery phase, so really focus on antibodies that will go into clinic as either a standard IgG or used as a model complex modalities such as multispecific or an ADC. But regardless, as soon as a sequence is cloned, we're one of the first people to assess it. And as you can see, there's a lot of different attributes that we're responsible for and have to determine with only 1/4 of a milligram of material. And we're actually working at the hundreds to thousands of clone scale. So really that kind of limits the types of instrumentation, really promotes us to really be able to scale everything that we do. But broadly, we're looking at three different attributes, three different categories of attributes. Some are related to accelerating the development process. So being able to use a platform manufacturing and not have to develop a special purification scheme can be really helpful. While other attributes are more geared towards meeting the target product profile, such as low viscosity, to enable a specific delivery method or dosing goal. So we really like to sit down with the project team to get a better sense of these target product profiles are. And of course, with the novel modalities I mentioned on the last slide, that takes a lot more careful attention since there's less late stage data on failure modes to keep in mind. But obviously, a lot of these properties are interconnected or interrelated. So if a molecule has non-specific binding, there's going to be another clearance mechanism. And so our pharmacokinetics and toxicity profiles are going to be impacted. And if there's evidence of low thermostability at this stage, we're not going to have a great time trying to conjugate as an ADC or even store it at late stage formulation buffers or concentrations. So always looking kind of like early stage indicators that something might not go so well later down the line. And the majority of these characteristics are being determined through in vitro assays. With a lot of tech development to increase the throughput of what we can do to mitigate risk and really be on top of letting our top candidates distinguish themselves. So I'm just going to go through a couple of different So I can't go through all these properties. So I just highlighted a few that we'll go through together. And again, this is going to be mainly from the early discovery perspective and how we deliver on kind of bringing forward the lowest molecule, the lowest risk molecule forward. And a lot of that work revolves around providing in silico tools and training so every project can be supported, particularly when it comes to immunogenicity, where something like a MAPS assay really can't be applied at this scale. And an in silico method is ideal. So Olga will kind of walk us through Immunoscreen, one of the tools that we can apply here.
Thank you, Tony. Next slide, please.
Mhm.
So I put this slide on. I don't need to tell to this audience of different factors to immunogenicity, but I guess my point was just to highlight that in silico tools would address only really protein factors like T-cell epitope content and not others. Next slide, please. So what majority of tools on the market and in pharma companies and biotech do, they tools which are used for preclinical assessment of immunogenicity. In silico tools focus on prediction of T-cell epitopes, which is considered to be a driver for high affinity B-cell responses, which leading to anti-drug antibody responses, and even kind of the tools really predict MHC binding, MHC molecules to drug peptides. They don't take into account actual binding to T-cell receptors and address only kind of a limited number of protein-related factors. The antigen processing is taking into account to some extent, but also not fully. Kind of with this limitation in mind, the next few slides are about how we use immunogenicity assessment in silico tools in AstraZeneca. And next slide, please. So Immunoscreen is a tool, how we call it in AstraZeneca. We start with MHC binding predictions, and for this we use immune epitope database tools, which we license. We predict binding affinity using NetMHC class 2 and class 1 tools, which there are kind of two varieties there. Binding affinity and eluted ligands, and we use eluted ligands tools which take into account MAPS data as well as binding data. We predict binding affinity to 27 HLA class 2 allotypes, which kind of suggested by IDB as providing good population coverage. And this includes DRB1, DRB345, DQ, and DP allotypes. For class 1, it's also about 27 HLA A and B allotypes. To decide on binders, we use peptides with affinity above certain thresholds as suggested by IDB, and for class 2 is 10th percentile, for class 1 it's 2nd percentile. Sophie, may I ask you to mute yourself because it's creating some kind of noise on the line, if possible. Thank you. For in vivo expressed therapeutics like CAR-Ts, they would use MHC class 1 screening as well as MHC class 2 screening. And next slide, please. Next step is using self-peptide filters. So peptides which match endogenous proteins, they will bind MHC molecules, but they will not activate T-cell receptors. So in kind of this step, we try to remove such peptides. We use concept of well, kind of self-like peptides. For example, for antibody type therapeutics, we would use human antibody germline to look at exact matches and to remove this binders from consideration for fusion proteins. We would look at endogenous human proteins, which are part of the molecule, and we will exclude them if there are exact matches. But we also look at the residues which kind of face T cell receptor, appearing human on a TCR facing residues, and here we match against a human canonical proteome. And on this little schematic, you will see that some kind of residues, residues 1, 4, 6, and 9 in the binding core, in the binding groove, they would look towards MHC class 2 molecule, and the others will look towards TCR receptor. So if the molecule, kind of the peptide, doesn't have to be fully human, if it still binds HLA class 2 molecule, but it needs to look human, just how the T cell receptor sees it. So we kind of look at the filtering self-peptides in this way as well. Next slide, please.
Quick question for you, Olga, on this.
Yeah, sure.
Facing. So are you using NetMHC Pan to tell you what the predicted core is, and then using that to then say which positions are facing to the TCR?
Yes, yes. So we use the core, and then they say here positions 1, 4, 6, and 9. They look into MHC. They could be non-human, but on the T cell receptor, it needs to look human. And we normally have maybe 30% of epitopes would be removed in that way using this filter.
Mm-hmm. And you mentioned germline and antibody constant domains. How much do you buy into use of OAS for, like, normally detected antibody sequences to use that as a reference, or do you only do germline?
In this case, we do only germline, but we use OASIS metric as a kind of first metric. Then we screen big libraries because it's much faster. So they use humanness as well, but not in this instance.
Mm-hmm. Thanks.
So next step is looking at allele frequencies for MHC molecules and calculating population coverage. I think we all agree that using allele frequency would allow for better population level risk assessment. We take these allele frequencies from allelefrequencies.net database. And we calculate the population coverage for each epitope and provide epitope maps, like one shown on the right, where you could see kind of what is the estimated population coverage from each potential epitope. And next slide, please. So what I forgot to mention, that we mostly focus on global population, but we have populations from different geographic regions, and if desired, we could calculate different population coverage. So Immunoscreen provides several metrics, summarizes all results in several metrics, and this includes epitope counts for different allele families, number of affected alleles, but also so-called immunogenicity score, which combines population coverage and DRB1 epitope count. We consider DRB1 epitopes kind of main focus of immunogenicity profiling, but we report the other allele families as well. Also, per sequence, we would provide an epitope map to show locations of all epitopes to identify kind of mostly epitope clusters and high-risk epitopes. And here they would be shown as shown in this kind of middle graph for DRB1, DRB345, DQ and DP alleles. Basically, you see for epitope starting at this position, how many alleles would be affected by this epitope. And this really helps if kind of project teams want to do deimmunization or germlining. This helps to identify where the high-risk epitopes are. And in the table on the right, you see the epitope counts and the immunogenicity score, how this looks like for example of these 4 antibodies. Next slide, please.
Sorry, Olga, one other question, because
Yeah,
this is
sure.
bugging me as well. I also see very high risk scores for trastuzumab, but we know
Yes.
it's not a very immunogenic molecule. What do you think's going on there?
So I think we always see, and maybe next slide is a good slide for that. We kind of see that, next slide, Tony, please. So what we see that humanized antibodies generally would have higher immunogenicity scores and higher number of epitopes. But also we see that human antibodies and humanized would have a huge overlap in the distributions. So then for antibody-based therapeutics, we would plot these distributions, and this is based on 217 approved clinical stage antibodies from Mark's et al. Paper. And we would think, where does antibody fit? Kind of in this case, if it's human, but it falls at the end kind of range of human antibodies, we would think that this is something to pay attention to. And the same kind of for humanized antibodies, if they fall very much into end of the high end for humanized, the range for chimeric, this would be the kind of flag. But of course, we know that sometimes there are very many epitopes, potential epitopes, and high immunogenicity score, but antibody is not immunogenic, and this would depend on all other factors like mode of action and things like that. And I will be talking more about that later.
Olga, if I may also comment to that.
Yeah.
For trastuzumab, we discovered that we have a very low DC uptake rate, and therefore also a very low presentation in the MAPS assay. Could it be also that we have here a nice example of why in vitro assays are still necessary, because we cannot assess uptake and processing with our in silico tools as of now?
Interesting point. Yeah. Yes, good to know. Next slide, please. So we looked at the performance of immunoscreen on these 217 antibodies where we know clinical immunogenicity. Well, I should point out that maybe because the data was collected by Marx et al. From literature in semi-automated way, maybe it's not a very kind of accurate representation of the immunogenicity. It all averaged for each molecule, number of clinical trials would be averaged, and there are different ADA assays and lots of differences. So kind of we shouldn't really fully trust this data. But what we observe, if you see the top graph on the right, correlation between number of DRB1 epitope counts and percentage of clinic ADA incidence in clinic, we observe kind of this very weak correlation, and correlation coefficient for that is 0.5, which is statistically significant correlation, but it's still kind of very weak or moderate. We do see kind of that the molecules with, if we bin the molecules by epitope count, as you see in the lower graph, that kind of high epitope count associated with higher percentage of immunogenicity, but obviously from the top graph you could see that there are lots of exceptions from that. And here we should remember that in silico tools take into account only some of the factors which contribute to clinical immunogenicity. So next slide, please. So how do we use it in AstraZeneca, this type of tools? The tools are implemented as a web-based tool, and we trained kind of scientists in biologics engineering, people who work on projects and early stage to run the assessment on their own. So it is kind of more high throughput. And I think last year they saw about 2,000 molecules run. So we really saw a huge increase in comparison than one person was doing manually the assessment. So mostly, most common application is at lead selection and optimization stage, where the project teams would select variants with low immunogenicity risk, use this information for to decide to engineer molecule or not. And they use it alongside other properties like developability, and they also do other activities. So they use germlining as a standard activity, and they try to improve stability of molecule, reduce post-translational modifications, basically improve developability properties, and immunogenicity is always used as a multi-parameter optimization, a part of multi-parameter optimization for developability. But also at other stages in early discovery, sometimes we use it to screen relatively small libraries, and we use it for assessment of different molecule formats. At the later stages, then candidate is selected. The in silico assessment would contribute to immunogenicity risk assessment document, which includes various factors of immunogenicity and answers different questions. It's a questionnaire type, kind of template type document. And there are questions, some questions would be about mode of action. In silico assessment would contribute about patient-related factors, clinical use, about product quality, and kind of potential clinical immunogenicity risks. This document would assign 3 types of immunogenicity risk: low, medium, and high. And this would be used to decide on the strategy for clinical immunogenicity assessment for the assays, which patients groups and would the placebo groups would also be measured or not. So next, I will pass this to Tony, who will talk about developability assessment, but maybe this is a good moment if people want to ask questions about immunogenicity assessment particularly.
Olga, thank you so much for again, bringing this all the tools and their application. And I'm very curious that when you started implementing it in your, with the discovery therapeutic group, uh, is there a timeline? Is it at the very, very early stage where you could implement, get the outputs, and still help them to optimize it? Like, was there enough time for them to go back to the drawing board and use your outputs to, because you said they're used in lead selections, and were they okay with just the in silico tools to give that guidance, and were not asking for doing some in vitro on top of it?
So in general, we don't do on regularly in vitro assessment, in vitro immunogenicity assessment in AstraZeneca, and it depends on which kind of project team you talk to. Some project teams were very keen to use this information, and they also had possibility for engineering, like further germlining. So they used this information and did that. Sometimes there was no need, and now we kind of getting much earlier into it, so we will kind of do it at hit selection stage. So these tools will be used earlier and earlier, and they start to get kind of more and more incorporated. And we're trying to improve the tools as well all the time.
Yeah, I can expand on that. So again, because it's such an easier to use web interface, people love to use it and just to kind of get that early stage data as soon as possible. And I have been on projects where they want to see the data pre-cloning to like really accelerate timelines. So that's kind of like another kind of benefit of having this so accessible, is that for the most part, all of our discovery pipelines are already humanized. So it's kind of a bit on the rarer side to see something kind of fall outside of that humanized to human range. But when we do see something like that, we'd probably do some sort of like germlining up front, or like kind of use the tool to like detect exactly the peptide that's kind of causing that signal to occur, and perhaps adding a variant that's like maybe a little less immunogenic. So again, you don't have to go through that another iteration of cloning and production. You have that kind of lower immunogenicity from the same backbone already kind of like in the same set to test against.
And the reason I asked you this question is that I'm happy to see that there's more embracing the in silico output itself, and not looking for further validations by doing in vitro assays. Because I remember 9-10 years ago, when we were trying to implement this, people were like, first show us by an in vitro or an orthogonal way that what you're saying by in silico is really true when you are rank ordering candidates, because you don't want to derail sequences just for the sake of epitope. So I'm just like, this is great that you can just use the in silico and keep on moving, rather than adding the in vitro to confirm at that stage. Yeah. Thank you.
Yeah, exactly. I think just overall of our de-risking strategy, just having a a more human sequence in the same panel, if they're both equal in terms of affinity and other kind of properties, we would definitely want to go with like the lower immunogenicity risk profile. And that's just kind of an obvious choice. So again, because their production is also kind of like higher throughput as well, we can easily just like add in that extra clone, and there's not really too much concern about that.
Okay, shall we move to maybe to give you time,
Can I ask
Tony?
a quick question?
Okay.
Hi, Olga. Hi, Tony. This is Dan from Amgen. What is your cutoff that you use it at? Is it the top 1%?
So our cutoff is based on this 217 antibodies published by Marx. We looked at the best threshold for kind of to explain clinical immunogenicity. We divide it into two classes, low and medium, because we don't like to say high from in silico point of view. And I wouldn't be able to disclose exact cutoff, but this is how we obtained it.
Okay, thank you.
Yeah, so maybe I can kind of bring us back to the developability assessment and how this kind of all fits into our kind of like overall like kind of risk assessment process. So again, like just want to highlight, again, people love to use immunoscreen, especially very early on. Again, to like accelerate timelines, to like really not have to go through kind of that reproduction phase if you do kind of find a hit. And then also you don't have to play the whack-a-mole game if during like early lead to optimization process. So you all have every single kind of like prediction in one go. And that's kind of like why we wanted to develop a pipe or a platform like Inside, to be able to like kind of package all that together into one tool to guide through both the early stage and lead optimization process. Inside is our, again, our pipeline. So it combines the front-end GUI where users can submit thousands of sequences, and then that all gets processed to create a structure from which biophysical features are then calculated from both the sequence and structure, and then fed into our ML models to predict developability risk. So again, we have a lot to keep in mind in terms of different attributes. So this is super helpful during our challenge of multi-objective optimization, and having some sort of warning sign of if we're introducing a new liability while we're trying to fix another one, particularly for some attributes that might be at odds with each other. For example, self-association at high concentrations is often driven by charged interactions. So to minimize that, we can introduce some more hydrophobic residues, but then at the risk of increasing aggregation. So kind of Insights allows us to kind of keep both considerations in mind while we're designing different sequence variants. And here I just want to kind of talk a bit about some of the pillars that really allow us to build and continue to improve on the platform. So the first being our full build out of our in vitro assay capacities for developability, particularly. And so this isn't really just pumping out more data per se. It's more a lot of like gathering a lot of late stage data to know if you've been running the right assay and if you're maintaining predictive power across novel modalities. But of course, all the assays do need to be scale and automation friendly to build ML-ready data sets. And the second pillar that we really highlight here is the expertise in AI/ML combined with subject matter expertise on the attribute of interest, which I'll go into in the next couple of slides. And then on top of all that is a lot of IT infrastructures that's being built. So thankfully, all the data cleanup is all streamlined and not really falling into just one person's lap. But of course, this is a continuous cycle, and we're always trying to improve those types of procedures and build out our models, not just for standard IgGs, but also for other modalities such as ADCs and multispecifics. So again, again, my main responsibility is developability, so I want to spend a majority of my time talking about that, particularly the high throughput data generation aspect of that. So that really comes into play when we're trying to build more generalizable models on a sequence space that keeps on increasing as we onboard new libraries and modalities are continuing getting added into the pipeline. So what's really great about our panel of assays is the low material requirement. So we're able to basically get a measurement of these different developability attributes on every single sequence that comes into us for early stage discovery, which is super helpful as we want to build our models to be sample a wide selection of the available sequence space. And we do see as we sample more sequence space and we're able to cover that wider distance that we're able to build more accurate models. And I will say we are kind of cognizant about the fundamental issue plaguing our clinical immunogenicity data sets, where various groups end up reporting data on what's supposed to be the same assay, but when you actually kind of dig into the methods section, there's actually a lot of modifications between protocols, which can make merging external and internal data sets a bit more challenging. So again, I'm also super curious to kind of discuss that further now that data as a service is starting to sprout up and how we navigate scaling up our data sets while not introducing new sources of noise from the source of that data. So this one slide can be its own full-length presentation, but I think my main point here is that to really build out a full suite of predictive models to enable multi-objective optimization, you really do need to take a lot of time from assay development and build that substantial trained test set. But of course, there's ways to build models when the nature data is a bit hard to come by.
Seema had a question around your non-specificity binding. What sorts of targets do you use for that?
Yeah, so we did spend a lot of time, like months, of really kind of understanding the different kind of attributes of each different antigen. So I think the classic BVP ELISA is a classic for a reason. I think we are trying to move towards a more reproducible antigen. So I think we're still kind of making that final decision in the coming month or two. But I do think we're moving away from BVP in some instances because we do see a bit of batch-to-batch variation, which can make, again, merging those data sets a bit more challenging. So that's one consideration we keep in mind. But I will say how we decide the antigen is based on PK data. So again, the point of non-specific binding is to kind of measure if there's going to be crashing out once you go in vivo. So we kind of base our decision making based on that type of data.
Thanks, Tony.
Okay, for sure. So yeah, so again, that in vitro data, particularly like I mentioned for non-specific binding, it's not always available to the scales that you really need. So here I'm just showing an example from some work from Joe Delmar from AstraZeneca, who built this very nice methionine and tryptophan oxidation model. And oxidation does seem to impact how immunogenic a peptide can be. So beyond the potential immunogenicity enhancement risk that comes from oxidation, we also care about this because it helps minimize batch-to-batch variation, which can also result in differences in potency in vivo. So for those of you who are not familiar with this actual oxidation process, a methionine residue first has to react with an excited state singlet oxygen. So that's often generated from photostress. So that creates this persulfoxide intermediate, which then can be converted to subsequent methionine derivatives. And the rate at which this process happens is also influenced by the formulation pH. So again, photostress is often what's causing this stress to occur. So what we do is to simulate this process happening over time during manufacturing and storage is actually to put our molecules through an accelerated version of this kind of photostress reaction. So we incubate them in our photo chamber for 7 days, and then we actually run these samples through a size exclusion column to get a sense of if any new aggregates or for fragmentation peaks have occurred. And then we'll probably do peptide mapping to detect if any of these new methionine derived species actually did form. And so if you ever had to collect this type of data, peptide mapping is very intensive, and that's something you can really do on thousands of molecules. So again, this is a somewhat constrained data set. But regardless, our data science team was able to team up with our analytical scientists to determine a number of key structural features that can describe this clinical process. And we were able to build a predictive model from a few in silico features. Here, this is a random forest model, so it is a bit more interpretable, and we did see that solvent accessible surface area was a pretty important feature, which makes sense from like kind of biophysical standpoint. And overall, the selected features were all reasonable, so that does bring a lot more confidence that our model has learned something about the underlying chemistry. And actually, last year there was a comparison of a couple of various PTM models on an independent data set. Which include this one from Jared, and did end up performing quite well. So it was just really great to see that this model has some staying power after it's been published so many years ago. But of course, there is still room for improvement. Actually, last month, there was actually a preprint published on tryptophan oxidation from a couple of authors from Genentech, which is very interesting, and actually pointed out that maybe local negative patches might also be an important driver for tryptophan oxidation. So again, a lot more features and kind of like expanding work around this field still going on. But then kind of zooming back into like their overall inside platform. Here I'm just showing some of the outputs from inside on the left. Is a distribution of the total number of developability flags that might come from a couple of hundred sequences coming directly out of discovery. So we can use this to help prioritize what sequences can actually get cloned and put resources towards characterizing. And now on the right is more of an example of a set of clones that what a set of clones might look like during down selection, where you plot all the different flags to select a subset to take a closer look at. In this case, all the antibodies looked pretty good, with only a few developability flags across the panel. But for this particular project, we wanted something on the lower end of hydrophobicity, and obviously a lower end, a lowest possible immunoscreen scores. So we're looking at this left quadrant here. And again, there's a lot of IT infrastructure that allows me to kind of generate these kind of graphs and access the in silico predictions that might have been done months ago, pre-cloning from someone at a different site, and then pair that with more in vitro data generated at different sites later down the line. And if I have any questions or want clarification on the exact method or anything like that on how the data was generated, I can reference a specific experiment date. And then so the scientists can spend less time kind of sorting through everything that was done for that project, and like we can continue moving on and making decisions quickly. But then expanding a bit on the IT infrastructure and what that looks like at
Real
AZ.
quick, Tony and Olga, just from that plot, I'm interested in how often does the Immunoscore like become a predominant predictor versus all the other developability metrics, whether measured or predicted?
Yeah, I mean, it's so product dependent. I would say, again, all of our discovery platforms are already been humanized. So I would say from that kind of point of view, it's very rare that we see something like very strange in terms of immunogenicity risk in general. And then we do see some differences in discovery platforms, so that might be a factor as well. And also the target, kind of like if something's super hydrophobic, you're going to end up having hydrophobic CDRs to kind of like be able to bind to that. So it really does depend on the target. And kind of like also kind of like the target product profile. So if we're trying to aim for a high viscosity formulation, that's going to be the number one thing we're going to look at kind of at this early stage. And we want to pick a candidate that's going to like meet that. So if there's any risks that we can't meet that target product profile, we want to just flag it as soon as possible.
Makes sense.
Yeah, and happy to expand as well if anyone that wants to do that. But overall, again, in terms of IT infrastructure, we did adopt gene-data biologics as our in vitro database. And that really has made great strides in making all of our in vitro data shareable. So I can just send back someone a hit list. And if I ever update that data, I'm not spending my time like kind of swapping back Excels or PowerPoints. It's all kind of just like on this one platform. So anyone can just download the newly pushed version. I will say, obviously, there's a lot of like kinks that go into adopting a new system, and a lot of work that goes into kind of putting all the infrastructure in place and integrating it with our in silico data sources, as well as our in vivo late stage data channels. And obviously, it's also not a one and done situation. So we're obviously adapting to new assays, like I mentioned before, new data sources, and also keeping track of all the different variations of very similar processes can also be kind of an interesting challenge, particularly when you're kind of like iterating on different structure generation tools or kind of like different ways to like make in silico features. All that kind of can also kind of get captured to compare against. But overall, this kind of data structure really has opened up a lot of doors in terms of the data-driven decision-making we can make, not only for the project-specific decisions, but also kind of answering broader platform level questions, kind of evaluating different risk mitigation strategies that we are considering. So something I'm really excited about is kind of the different mindset we're having when we're selecting a different or the best data set to answer a question. So I think previously is a kind of a limitation that whoever's kind of like designing the experiment might reach for whatever molecules people are familiar with or had easy access to. And that ends up just going into the experiment. But now, because we have easier access to our entire wealth of data, we can kind of limit that bias to like reach for whatever the easiest to get to. And now we can just filter based on kind of the desired criteria and whatever associated metadata we're looking for to create a subset, and then have that hit list go forward for production, and really kind of like combat kind of like the bias we're seeing, kind of like avoiding negative data. So again, when we design an experiment, we have a good mix of both positives and negatives. And that really does, so again, kind of going back to like a lot more attention being shifted towards experimental design. And it does result in a lot of gains in terms of how data is recycled across projects and therapy areas. But I will say one of the highest demand types of data we have are sequences that have late stage outcomes attached to them, particularly clinical ADA, which Olga can share some of the early data exploration work going on with that type of data set.
I was thinking maybe some questions on developability now.
Sure.
Yeah, can I? Thank you so much. This is really exciting. Can you share a little bit about the in vitro assay measurements? Like what kind of in vitro assays are being used for just to do this integrated analysis?
Yeah, I mean, so this is kind of an overview of kind of like the different attributes that we predict again at early stages. So again, we're quite limited in how much material we can get our hands on during the first production scale. So like, again, it's very small. We're only trying to produce just enough to be able to answer the questions we have. But this kind of covers a bit of the kind of different attributes. So again, for non-specific binding, that's a pretty ubiquitous concern we have. We want to make sure all of our targets or all of our antibodies are just interacting with the designated target. And then reversal self-association is more for, again, predicting that kind of like high viscosity risk to be enable like kind of subcutaneous delivery. So again, we would kind of prioritize different developability attributes depending on that target product profile we're looking for. So if the product or if the team isn't really looking for that type of kind of formulation or delivery method, maybe we would kind of like deprioritize that type of attribute, and then kind of like bring in more of the other ones. But yeah, I think this is a pretty standard list that I've seen across a couple of different papers in terms of like everyone does a very similar kind of panel to look at anything that's kind of just kind of like not be able to really make it through the development process.
So these are developability assays. They're more analytical characterizations of of the actual drug product, right? So these are a part of that. But what we have seen also from our agency perspective is to ask, like, can they change the biologically relevant risks, right? So for example, a lot of work was done initially on some of these parameters, like hydrophobicity or pH based changes, or even colloidal instability. They can cause particulates, and you mentioned tryptophan oxidation, for example, that causes hot spots or misfolding, leading to like subvisible particles, aggregation, in some cases, just um, uh, like building a new neoepitope, which can become a problem. So, have you connected that to those parameters, to some of those innate impurity assays that the agency has been asking for, or controlling thresholds of those parameters, which will relate to the risk later on? I asked that question because you were showing up a lot of immunoscore and the developability, and I wasn't sure what the immunoscore was scoring. Was it just the initial in silico intrinsic risk, or was it also bringing these post-translational risks that come from all the factors that I just mentioned?
Yeah, I
It's
mean, that's
just
a
intrinsic.
Olga, you wanna?
So it's just intrinsic in silica risk. There be one epitopes which contribute to the sequence to give the population frequencies of allele frequencies.
But would you be looking at those additional post-translational risks, which are analytical characterization? I understand the assays, I do, but there's also a request now from the regulators to add that to your PTM-based risks, to look for innate phase assays, to look for priming of the innate to adaptive, and then how does that relate to the changes in these parameters, the CQAs?
I don't think we look at it. We have been looking at these assays, or if we look at them, we would be doing it much later in the well, not in early discovery. So it would be closer to kind of clinical studies, and possibly there are programs where they looked at it, but I don't think me or Tony are aware of any such examples.
All right. Thank you so much.
Yeah, and I can kind of expand. So again, like we do kind of also do peptide mapping. So like if there is some sort of oxidation risk, that would kind of be flagged early on, kind of before we continue on with development. So we can either do some sort of like lead observation if the candidate's like kind of like very superior to like in other kind of attributes. So that could be one thing we do. And again, that's kind of like the power of inside, where if there was that type of like oxidation risk that ends up happening, we will to kind of do some sort of sequence variants while we can kind of juggle all the other kind of different attributes that the molecule already kind of showed promise in. So yeah, I think kind of the direct measurement we would do is like comparing immunoscreen and actual peptide mapping data. I think the kind of, again, like you were mentioning, the sub-visible particle, that would be a bit later down the line, but we would directly measure the actual oxidation rate. So hopefully that would be like very close to zero. So we won't even have to worry about the kind of like the downstream issues that also might come from that.
Got it. Thank you.
I'm happy to take any other questions on the developability section.
So maybe I just use the last few minutes to talk a little bit about clinical immunogenicity prediction, basically our attempt at doing that. Next slide, please. So what we try to do to put together data on molecules which went into clinic and have clinical data on ADA incidence, and we gathered database of around 260, mostly external molecules, but some kind of internal molecules, and we try to add all these missing factors of immunogenicity, causing immunogenicity. So we have protein factors, which is in silica epitope content, and we try tried to add in silico developability properties calculated from sequence and structure, kind of with 3D structural descriptors as a replacement of factors from product. And we added information on the treatment and patient, factors looking at the target for molecule modality, route of administration, and patient population immune status. And we used machine learning and AI methods to try to build a predictive model for clinical ADA incidents, with the idea that this would allow us to rank molecules much earlier for risk of immunogenicity and to enable kind of lead selection for clinical immunogenicity prediction. So next slide. So here is a little bit more information, what features went in, and maybe I just comment on the in silico developability properties, which were calculated from homology models built by AlphaFold 2, which accounted for 730 features. And here we removed some of the kind of highly correlated and low occurrence features. And overall, we had 750 features accounting for all these factors. Next slide, please. Actually, we will skip this slide. It's just about dataset content. But I want to talk about modeling approaches, which we used. So we kind of had these 3 buckets of different types of features, but also we wanted to use protein language models, model embeddings, and Sapiens, which kind of, I point here to the paper where it's described, worked best, and we used the principal component analysis in kind of this space as well. And then we used classical machine learning methods on top of that, and we tried classification and regression methods, a variety of machine learning algorithms. Next slide. So results were kind of disappointing. So it's almost a negative attempt. So here we kind of didn't see how much added by in silico developability descriptors or the features which are responsible for target, route of administration, format, this type, because models built on one descriptor, which is total epitope count, from Immunoscreen, they're actually similar to models with many descriptors. And protein language model embeddings also didn't really improve accuracy. So our best model, regression model, had R-square of only 0.36, which we didn't consider to be really useful, and we kind of, at the moment, we are continuing with Immunoscreen measures to kind of assign risk of immunogenicity to molecules. But what we thought here, we had such a noisy endpoint, percentage of ADA per molecule. And here, I think what you done with your database, it's a really huge improvement, because you go with cohort, with time point, and this really provides much better kind of definition to this data. And possibly our model was lacking important features, or the features were not kind of high quality enough. So I think the databases on clinical ADA incidents would be kind of very useful for everyone for future modeling efforts. And next slide. Well, that's kind of, I think you all agree with that we cannot yet predict clinical immunogenicity. And next slide has kind of our colleagues who participated in the development of in silico immunogenicity and developability tools. Thank you for listening. And sorry, we kind of almost overrun.
Thank you both so much. Yeah, we do have time maybe for one question, if anybody has a question.
So if you still need to add one in vitro assay on this, which one it would be?
If we add what, sorry, in vitro assay?
to improve the in silico model, if you want to add one in vitro assay
Okay.
in terms of uptake or T cell or activation?
Well, I would add looking, because we have whole molecules I would add kind of T cell activation proliferation assay for all these molecules if I had one. And ideally from the same provider.
Thank
Not
you.
biased at all by your T cell antigenicity federated learning effort.
Well, this is different. That's for peptides. Yes.
So I had one comment. I think all of the antibodies that you looked at, we know now, especially if there are bispecifics in there and immune activating molecules, we know that there is non-sequence based risks that we're just uncovering. So if you were to go at that list of molecules and remove all those that had potential non-sequence risk, would your score get better? Would your R squared be better?
Well, I don't know. We didn't try that because whole idea was that we bring in all these additional risks in and model using these risks. So there were bispecifics, and the mode of action was such that it put them in potential risk, but we kind of used these features. So our hope was that this would improve prediction. But I will try kind of what you suggest. Yeah.
Yeah, but I mean, even looking at your in silico score, because that's just sequence based. If you've got something that's non-sequence, it's not going to correlate. There's no way.
Yeah.
Right? Yeah. But very interesting. Thank you, guys.
All right, awesome. Well, yeah, thank you both for taking the time to prepare these slides and to field all the questions and facilitate the discussion. This was really great.
Thank you for inviting us. Yeah, it was a pleasure. Bye-bye.
Okay. Thank you all.
Thanks, guys. Bye.
Bye.